4 out of the 7 vaccines are still egg-grown. Pregnant women is not a separate population that should be tested in a separate study. They need to manufacture new vaccines every year. We haven't seen a lot of breakthrough in the technology, which is a bit surprising. In Europe as well as in the US, you need to have a specific pediatric investigational plan that you need to discuss with health authorities.
Hello, this is Industry Voice by OCT Clinical. The topic of this episode concerns the area that has been given an additional limelight starting 2020, vaccine development. With our guest today, we will go deeper into challenges and pitfalls of this research field. Please meet Alexander Breidenbach, Chief Development Officer at ACM Biosciences, the leading Swiss-based biotech company.
Dear Alexander, thank you very much for being with us today. It's a pleasure to have you.
Thank you very much, Anna. It's a pleasure, and thank you for the invitation. It's great to have the opportunity to talk about a very important topic, vaccinations. It's in all our mind at the moment. Thanks a lot for reaching out.
Thank you very much for coming. So diseases that once burned through populations are now increasingly rare because of vaccines. Million cases of childhood diseases are being prevented today through vaccination. For instance, measles, mumps, and rubella. How would you describe the change in the vaccine development industry over the past years? And what are the current trends in immunization?
Thank you for the question. I think it's a fantastic start. And, I think we all agree that vaccination or the discovery of vaccines is an unprecedented success story in medicine. Vaccines are so successful that we all feel they are normal. They are with us. Every one of us has, experienced vaccinations as children, as we were child, and later on. So we all trust in the safety and efficacy of vaccines. What changed now since 1.5 years is that suddenly there is a vaccine that is threatening the world, and that is much more different than when we talk about measles, mumps, rubella, and diphtheria. Here it's threatening vaccines that also needs a differentiated response to those. So now in the last years we had something like HIV, we had bird flu, we had Ebola, and now SARS-CoV-2. But with the previous pandemias or these outbreaks of new viruses, and also with bacteria threatening the safety of many, many people, we haven't seen a lot of breakthrough in the technology, which is a bit surprising. So what we've seen in the past was new technologies. We had genetic engineering that actually replaced the more empiric development of vaccines. But when you look at the flu vaccines that are available for the 21-22 season in the UK this year, interestingly, 4 out of the 7 vaccines are still egg-grown. So you take chicken eggs, you inoculate it with viruses, and then you harvest the viruses then and make vaccines out of that. That's not a very sophisticated technology. And also the point is, I would say it takes long. It takes much longer than what we need if we need to tackle a very serious disease. And this is in an environment where there is a huge market. There's a huge market and there's a huge unmet medical need for vaccines still outside of SARS-CoV-2. So flu vaccines, for example, they make about $6 billion in 2021. And it's still based on outdated technologies. We have other vaccines that are important in our life, like human papillomavirus vaccines, pneumococcal vaccines, shingles vaccines. It's huge markets, but at the same time, the technologies didn't advance as much as the medical need exists. This has changed tremendously last year, as we all know. mRNA vaccines are new, and they were fast. They were fast in helping us actually tackling with the pandemic that we are all experiencing right now. But the biggest surprise was, because mRNA is not a new technology, mRNAs are around for many, many years, but to develop the vaccine on an mRNA basis within less than a year, that is fantastic. That is absolutely shows this technology is changing the world and is changing the world of vaccination, and it's a revolution. But still, you know, there's a lot of deadly viruses and bacteria out there where we need to have new vaccines. And since we don't have them yet, with the mRNA vaccine technology, that might actually change now. That's good news for us. So the current trends in immunization, to get back to your question, I think it is clearly mRNA-based. It is based on new delivery technologies. And with that, I think I'm very optimistic that we will see more successes in that field.
And are there any challenges in implementing scientific advancement to improve effectiveness of vaccination as a primary preventive strategy against infectious diseases?
Yeah, that's a very good question, in a sense that you know, there's three major topics to address, maybe, that all hang together. There's of course the efficacy, but there's also the safety, and there's the manufacturing topics. So the efficacy of existing vaccines is very, very good, you know. So the point is rather, what is the magnitude of the protection that we achieve with the vaccines, and the duration of the protection. And here, I think we need to look at this case by case. For example, we all know now that the mRNA vaccines for SARS-CoV-2, that they need boosting because the protection goes down over months. This has to do with, on one side, upcoming variants of concern that are more, you know, faster spreading and have higher transmissibility. But also with the fact that we don't know much about the mRNA vaccine and the durability right now because they are so new. But overall, I think this is something that also needs to be better understood. And certainly what we all want as you know, recipients of vaccines is that we have something that provides longer efficacy, longer protection, maybe even lifelong protection. So one prick in a lifetime against a certain virus would be certainly more wanted than every half a year getting a booster vaccine. Same is true for flu influenza. Every year, the WHO, as you know, announces the strains that might be likely coming over to the Western Hemisphere and will dominate the next flu season. And by that time, the vaccine developers, they need to manufacture new vaccines every year. So this is certainly something where getting a universal vaccine for all flu variants and strains would be certainly very, very desirable, which provides protection for life. So I think these are challenges we see with the efficacy, but of course there are also challenges regarding delivery. We've seen that mRNA is fantastic from the modality to treat a disease, or even to provide immunity against virus diseases. But on the other side, the delivery is a hurdle here, because they need to be encapsulated and protected against the body own system that degrades RNA very quickly. So they are encapsulated in lipid nanoparticles. And we all seen that this is quite complex, and it's very unstable and requires freezing temperatures for storage and even potentially for manufacturing. So this is also something which shows room for improvement for the delivery, which also ultimately also hangs together with safety and also with efficacy. So more targeted delivery technology that can be easier manufactured is certainly needed here. These are the challenges that I see.
Well, thank you very much for the feedback. And you already started talking about the COVID-19 vaccine, and I wanted to go back to that topic. So we have always believed that vaccine development can take anything between 8 to 10 years. But today we see so many breakthroughs. Some companies manage to develop COVID-19 vaccines within a very short period of time. What do you think, what is, in your opinion, the shortest term for creating a vaccine without compromising quality and safety, of course?
Yeah, no, fantastic question. And I think everybody is now, looking very surprised about the timelines that we've seen. We all know that vaccine development in the past took a long time. But as I said, it was because of maybe not much progress in new technologies. When we still do flu vaccine development in chicken eggs, I think it shows that this is certainly, I would say, a last century technology which is outdated. But the corona vaccine development based on mRNA technology showed us how fast actually we can be. Will it be in the future less than a year, as we've seen with the Pfizer and the Moderna vaccines? Certainly not. I think a pandemic that threatens the health of the world population will again show very short timelines, but I think in routine development measures, I would see that we are not going back to 8 to 10 years. I could rather see we are going back maybe to something that is 5 to 6 years. Why is that? I think on one side, the technology has advanced and we have new modalities now that we can use. We still need to go through phase 1, phase 2, phase 3 development. That takes time for sure. And we don't want to compromise on quality of safety. That's also for sure. But there's also movement with the regulatory authorities. They look at platforms and platform technologies nowadays with a different view than in the past. Now you can actually use a lot of information you have from past development programs where you use the same technology, the same platform, and you had tested it already in humans. You don't need to start from scratch with your development activities. This will save time and resources. And with that, I believe that 5 to 6 years could be something that is realistic for future developments of vaccines. But exceptions might be possible as well.
And how long does it take to develop your vaccine? And can you maybe share some results and the next steps?
Yeah, so to develop our vaccine is not yet the case. So our vaccine is now knocking at the door of phase 1. So we have our IND package ready. We are in the last activities of our manufacturing process. So we are very confident that we can start in Q1 our phase 1 with a corona vaccine. The difference here is it is a beta variant of the spike protein. So we don't use the wild type. All the existing licensed vaccines are using the wild type spike protein. We use the beta vaccine. So the timelines are a little bit getting back to normal. We still hope we don't need 5 to 6 years. We will be fast and we will be hopefully getting the support from regulatory authorities to shape our development program such that we can move swiftly through the later stage development. So, but the difference of our vaccine is that with the beta strain spike antigen, we see very good cross protection in our animal models. So cross protection against other variants of concern, including the Delta variant, which makes us very, very hopeful that we have a very good vaccine and hope to see the same data also in humans. And given the rising numbers that we see at the moment with COVID-19 infections worldwide, we believe we also need a next generation vaccine, protein-based with a different spike variant than the existing vaccines.
And does ACM Biosciences take into account the ability of the coronavirus to mutate?
Yes, it's of course a very, very difficult question because we all see almost on a monthly basis new variants popping up. And then the question is, are they more infectious? Are they more dangerous? Do they escape the immune system? At a certain stage, you need to make an informed decision here. We decided to go with the beta variant because of the features of the spike protein that is very much helping to protect also against other variants, existing variants of concerns. Of course, we don't have a crystal ball. There might be other variants of concern emerging for which the protection of our vaccine and maybe also of other vaccines will be less so. However, as I said before, we are in contact with regulatory authorities to familiarize them with our platform technology. And with that technology, we believe we have a plug and play system, i.e. we can remove the mRNA, the protein construct that we currently have, and integrate another one of a new variant of concern very swiftly. So we don't need to go through the whole preclinical development and can actually bridge a lot of these studies and go swiftly into clinical development. We could do this with protein, as we do right now, but we also could do this with mRNA. But at the moment, we are working with a beta variant protein variant.
And now let's talk about specific populations, pregnant women and young children. Do you approach clinical trials differently in these cases?
That is a very good question. I would maybe put it into two pieces. Let's maybe talk about first about pregnant women. I think the immune system in pregnant women and also the tissue, the immune capabilities of the placenta, for example, of the uterus, as well as maybe in lactating women, in breastfeeding women, the breast itself is actually via the evolution best suited to protect children either nursing or being still fetuses against threats from the outside, against antibodies, because the antibodies that will be developed under vaccination will be transmitted to the fetus and protect them. So we have interactions with health authorities where we actually discuss exactly that topic, saying, should we have separate studies with pregnant women or not? And at the moment, I think we are in a situation where we anticipate a consensus that pregnant women is not a separate population that should be tested in a separate study. In contrast, pregnant women deserve the same speed of protection via new vaccines as the other population, adult population. So that's our opinion. Pregnant women should be included in the regular studies as well. It's different for, maybe back to the pregnant women. Of course, the preclinical package needs to provide the safety for testing in pregnant women. It means, of course, the complete reprotoxicology package need to be completed and need to be safe before you could do such a study. On the other side, when you talk about children, of course, children is a different topic and needs very careful examination and considerations before. So as we all know, in Europe as well as in the US, you need to have a specific pediatric investigational plan that you need to discuss with health authorities. And I think there are specific studies, sometimes needed, sometimes not, but you need to prove also in preclinical studies the safety in juveniles before you can actually test in children. But first and foremost, you need to go into an adult population. You need to go down as low as 18 years of age. And then once you have established safety in that population and the efficacy, of course, then you can think about and consider and discuss with health authorities and ethical committees the testing in younger populations than 18 years of age.
And with that being said, I think we can wrap up the COVID-19 topic and move on to the general vaccine development questions. So there are numerous tools that have been recently developed to help accelerate a vaccine development. Novel technologies and platforms are emerging. Could you name some of the most meaningful ones?
Yeah, so I already talked a bit about the old technologies, and just as one representative, I see the inoculation of chicken eggs as something that is last century technology. It's still used and it still has its value, certainly, but I believe that new technologies clearly are here and available and should be used. But within these new technologies, there are some that already showed its limitations. When you look at virus vector systems, for example, also used in the corona space, they are very good. They provide very good protection. But on the other side, they have the limitations that repeated administration is a difficulty, because these virus vectors, they themselves lead to neutralizing antibodies, which limit their applicability if you use it more than once. And we see this already now, that that in Corona, the booster vaccines for boosting, you will not use, in most cases, virus vectors. Then on the other side, you have other technologies like, using proteins, recombinant proteins, and also combine many antigens in one to have actually a broader protection against a multitude of strains that might pop up within the same virus disease. DNA vaccines, they have their challenges. As we know, it's very difficult for them to enter the cells. They had some setbacks there. I think what is most promising at the moment are, on one side, the mRNA vaccines that are encapsulated in lipid nanoparticles, but there are also other delivery systems that are very promising. And one example is the Novavax protein vaccine that uses nanotubules where the protein is fixed to when it's administered. But there are also other encapsulation technologies, for example, our polymerisome technology, where we encapsulate either protein or mRNA entirely into a vesicle, that it's only delivered to the cells where it can exert its effect. And that's antigen-presenting cells, in our case, where protein and also mRNA can be delivered to. And I think these are novel technologies. They will make a huge difference in the future for development of novel vaccines.
Thank you very much for such a thorough answer. I think our audience will learn a lot from this. And now moving on to clinical trials. So today, more than a half of all clinical trials are being outsourced to CROs globally. Do you outsource to CROs or do you perform the activities in-house?
Yeah, so before I joined biotech, I was actually in large pharma for many years. And be it biotech or be it large pharma, I think it is standard, it's industry standard to closely work with contract research organizations, be it for preclinical development or be it for clinical development. It is mandatory, it's you have very sophisticated, very experienced and top-notch and state-of-the-art CROs worldwide. And we are happy to outsource our studies to those CROs and to collaborate with them closely to deliver on our objectives for our program developments. And with that, it is absolutely clear that it's very difficult, in particular for small biotechs, to start in-house capabilities. We have maybe one or two clinical programs at the same time, and it would be a huge burden on our resources to have these in-house capabilities. What we have is, we have the core expertise, we have the clinicians and the researchers who have all the know-how and the knowledge, and we use and collaborate then with contract research organizations to use their capabilities, their expertise in executing on our development plans. And we've seen that this in the past worked out very nicely. It is a win-win situation for both situations. We save resources and we have capable partners on our side.
And could you please maybe expand on your CRO selection process? How do you approach that process?
Yeah, I would say this is maybe based on, let's say, my experience, and it might differ from others. But I would say clearly networking is very important. For me, what counts is experience with a particular CRO in the past. Many people in biotech come from big pharma, so they have past experience, and that helps. If you have existing relationships, then you can actually start on a much higher level and being much more efficient than if you start a relationship from scratch. So this is something which I count as a very high point. Knowing each other is important for me. The second thing is, of course, speed and service is of the essence. If I, as a biotech, am seen as a small partner that is not important and I will be on the back burner of a CRO, that's certainly very important for selection process. I need to feel like being handled like a big pharma entity, despite the fact that I come maybe with one program. while Big Pharma has 10 programs at the same time with the CRO. So a service that is tailored to our needs is important. And also, obviously, budget is an important issue for us. So a tailored budget that we can afford, and that is not seen as, a big pharma budget, that is also important. And the last component that I would say is worthwhile to mention is quality. So while the budget is important, it is not the utmost important criterion. We would not compromise on quality.
And on a closing note, if we talk about the future of the industry and research, what trends do you anticipate?
Yeah, this is a fascinating time we are in. When you look at recent developments, and it's not just Corona-based, it's also the whole revolution in the gene therapy, gene editing, RNA therapeutics, RNA editing. There's so much going on, and it's not yet the end of the story. At the moment, on an almost weekly basis, you see new biotechs being created, being funded, that explore that field, being it epigenomics, being it epitranscriptomics, to name few. They hold so immense promise to target diseases that we were never able to target before, And that is based on the possibilities you have with these technologies nowadays. In former times, when we talk about small molecules and even monoclonal antibodies, genomes, they just encode for about, I would say 1% of the genome encodes for proteins, or 2%. And out of these proteins, only a fraction of it can be targeted with small molecules and monoclonal antibodies. Now, going to the root cause of many diseases, be it the gene, or be it the mRNA or other RNAs, you have so much possibilities. I think the future of medicine and pharmaceutical development is unlimited. It's so bright, and it's the good news for patients in need, because we will be able in a couple of years to tackle many more diseases for which we had no angle to start with. Now we have these tools, we have the research, we have the experts, we have the funding, that I think we can make a big difference in a couple of years for many diseases that are horrible, that have a high unmet medical need, and also it will be at a different price angle. That's also what I believe, that we can also deliver it to many patients in the world, be it in high income or low income countries. That's a view that I have, and it's a vision that I have, and that's something that makes me excited being in pharma industry and in biotech, and doing the research and the development in areas where we can actually serve these patients.
Alexander, I think we can wrap it up here. That was a very productive conversation. Thank you very much for sharing your thoughts. I wish you all the best.
Thank you, Anna. It was a fantastic meeting. Thank you for your interest, and thank you for the very nice conversation here. Looking forward to moving ahead with maybe you and with other partners to deliver on our vision.
Let's hope we can collaborate in the near future.
Thank you.
Thank you.
Thank you for watching. If you would like to be our next guest speaker, reach out. You can find the right email in the comment section below. And don't forget to subscribe.